All right, class, so this is for what we didn't finish in class, going over the first part of the chapter 10 notes. We last left off drawing the neuromuscular junction and labeling all the essential components that you need to know. So the neuromuscular junction, it's where a motor neuron meets a muscle. And usually, you can see here in this picture, this neuron is meeting usually in the middle part of the muscle. And the synaptic knob of the neuron is in that junction, what's called the synaptic cleft, C-L-E-F-T, and then the motor end plate are all the components of the neuromuscular junction. And here's a closer picture of the motor, this is the motor plate, this is the synaptic knob, and then here's the cleft. Within the synaptic knob, there's these vesicles called synaptic vesicles, and they're filled with the neurotransmitter is in the blank there, called acetylcholine, and abbreviated ACH. And you can see over here in the picture, when calcium pumps in, when the message gets to the end of the neuron, okay, that changes the electrical environment, okay, and that's going to cause the vesicles to release the acetylcholine into the synaptic cleft. So the acetylcholine has the message, the chemical message, and then it's going to go to the motor end plate, the end of, or the beginning specialized region of the muscle cell, the sarcolemma. It has numerous folds within it, and these folds have these acetylcholine receptors like we drew, and they are triggered by the binding of acetylcholine. So acetylcholine will come bind on those receptors and allow sodium to enter, okay? And also, we haven't got to this point in class, but it also later will allow potassium to exit through different channels. The space is between the two, like I said, between the motor neuron and the muscle cell. And you're going to love this word. In the synaptic cleft, there's an enzyme, and that enzyme is called acetylcholinesterase. Acetylcholinesterase. And that enzyme hangs out there, and that enzyme's job is very specific. Its job, so it's an enzyme that breaks down acetylcholine. So once acetylcholine is in that synaptic cleft communicating the message from the neuron to the muscle cell, well, you don't want the message to be forever, so that message has to be erased, and acetylcholinesterase does that. This is a slide that talks about what's called the resting potential. So remember how I said salty banana? Outside the cell is a lot of sodium, so positive environment, and inside the cell it's potassium, or a negative environment. I don't know if you can see on this image, but check it in your book if you want, but it says RMP, or resting membrane potential. That is important to be maintained for a muscle cell to be able to respond to a signal from the brain to contract. So it needs to be maintained at about negative 90 millivolts inside the muscle cell. And it's set by leak channels and pumps, okay? Sodium-potassium pumps is the word here. So it's maintained at negative 90 until a message is received. That's called the resting membrane potential. So that is the end of 10.2, and now we're going to talk about 10.3. So more details about the neuromuscular junction and what else happens to make a muscle contract. So here, we've looked at this picture before. We drew out where a neuron meets a muscle cell. And you need to know these steps, okay? There's a lot of little pieces that are happening, but the overall, how does a muscle fiber contract, is a big process with many little steps. So the neuron has to excite that muscle fiber. Calcium goes in to the neuron, opens gated calcium channels. Okay, is the blank here. Calcium goes into the knob like we drew and lets the neurotransmitter out of the vesicle. Okay, and then acetylcholine goes into the space via exocytosis. So here's our synaptic knob, calcium's going in, neurotransmitters going out. And acetylcholine then, the second step, will bind to its receptor. Receptor is the word here, at the motor end plate. And then it excites the muscle fiber. And now the electrical message, the end plate potential, is traveling now on the muscle cell. Because it starts spreading because sodium diffuses in through those openings, those channels. And the cell briefly becomes less negative because this has more positivity going in it. And then the message, it's the start of the message on the muscle cell to contract, and then the propagation is the word there, propagation, along the outside of the cell called the sarcolemma, and it dives into those T tubules. And when that message is spreading, it has a specific name. It's called depolarization. So sodium goes in. The membrane depolarizes, and then right after, the membrane has to repolarize or go back to resting state. The action potential actually reaches what's called threshold, 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 and then the message starts. Okay, oops, back. This word here is called depolarization. And you can see the word right there. The cell depolarizes, becomes less negative. Eventually, it'll let so much sodium in that it becomes plus 30 millivolts. And just after sodium gates open and the message propagates, the message has to go away and it has to go back to resting. And how it does that is it repolarizes. Repolarizes. And how it does that is lets potassium out, and then it returns back to negative 90. When the muscle cell is depolarizing, or sodium's going in, and repolarizing, potassium's going out, it is in a, how do you say, I can't remember the word right now, but it's unable to respond to another stimulation. It's essentially can't respond. I forget the word. I'll have to look it up later. Just ask me. All right. So once that message is on the muscle cell, it'll eventually reach the sarcoplasmic reticulum. This is the cisternae. Same thing. SR cisternae. And the action potential will open those voltage gated calcium channels, and they're going to travel to the actin, and they're going to cause the troponin to change shape and move the tropomyosin over out of the way. So move tropomyosin, move here. So actin is exposed. And what happens when now the actin is open, now the myosin heads can bind and pull. Now, that is called the myosin heads grabbing the actin and pulling them, dropping, grabbing another spot, pulling, dropping is called the cross bridge cycling. And there's a nice picture in your textbook. Essentially, that picture should look like this, and these are the steps of them. Step 1, the myosin head attaches to the actin. Step 2, power stroke. Okay, the myosin head pulls the thin filament towards the center. When that happens, energy is being used, ATP, and it's released what's called ADP plus phosphate. If you can think of, ATP as adenosine triphosphate, and it's going to look backwards on your screen, I'm sorry, but I wrote ATP triphosphate. When it's used, when that energy is consumed, it's split into two different molecules called ADP and a free phosphate, and it releases that. That's what that means. And then the third step, okay, so it grabs, pulls, and it drops. It drops the actin. And because it drops it because an ATP binds to it. So a new molecule of energy. Fourth step, ATP is split, and essentially that myosin head is been cocked, like you cock a gun, and it's ready to fire again. It's ready to grab actin again. So the ATP provides energy to cock the myosin head. Now, what I would do is I go through each step, use your arms to demonstrate what's happening, and talk yourself through the cross-bridge cycling. All right? And that's what's actually causing the filaments to slide across each other. This will happen, the myosin will grab the actin and pull it as long as calcium and ATP are present. As long as the calcium has moved the binding sites out of the way, and as long as that muscle cell has access to energy, ATP, and it will result in the sarcomeres shortening, the Z discs moving closer together, and that's a muscle contraction. Now, you should note that the thick and thin filaments remain the same size. Okay, here's my myosin, here's my actin. All they do is they slide past each other. They don't actually shorten themselves. They slide past each other, past one another, the filaments. The sarcomere shortens, but the actual filaments do not change size, do not change shape. It's referred to as the sliding filament model. And there it is. Okay, so there's our Z discs. The muscle's relaxed. Now we're going to contract that muscle. Whoop, sarcomere shortens, and the myosin is grabbing the actin. There is a few things of interest when we're talking about muscle contraction. One of them is called tetanus. Now, tetanus can be caused by something called Clostridium tetani, a bacteria that lives in the soil. This is why we get tetanus shots, to prevent this. And if somebody gets tetanus, what is happening is that bacteria is blocking the inhibitory neurotransmitter in the spinal cord. That's going to cause contraction of muscles, complete contraction of muscles. So essentially, non-stop access to calcium. So the body's in a continual state of contraction. The muscles aren't allowed to relax. If you've heard of the term rigor mortis, or a body goes stiff, essentially that's tetanus to the extreme. The muscles are totally contracted, stiff as a board, the body is. Botulism, also caused by a bacteria toxin called Clostridium botulinum, and it prevents the release of that neurotransmitter at synaptic knobs. It can be very harmful, but also this toxin can serve some good purposes. It actually can treat a disease or a condition called cerebral palsy, where there's spasmicity, I'm not saying that right, in the muscles. It can calm them down. And it's also used to diminish wrinkles in the face by calming the muscles out, essentially calming them down. Okay, so we've talked a lot about how the neuron tells the muscle to contract and how those actin is pulled closer together to the Z, and the Z lines shorten. And tetanus is an example of when your muscles are continually contracted. Well, you don't want your muscles to always be contracting. It's going to be exhausting.